Hello everyone and welcome back to the session. In this session we are again discussing some important question from the NEET point of view, and in this session we are going to discuss about the chapter that is respiration in plants. As we all know, as the human being respire and undergo the various breathing exchange and the breathing cycle, like in the same manner plant will do the process. For example, the various kinds of respiration. For example, it will be very much sorted with the help of the questions which we discuss in the session one by one. Let's start the session. The first question from the session is RQ, that is the respiratory quotient of protein, carbohydrate, fat and organic acids are in order. Option A, more than 1, 1, more than 1 and less than 1. These all are the sequence for protein, carbohydrate, fat and organic acid. Option B is less than 1, more than 1, 1 and 1. C is 1, 1, 0 and minus 1. Option D is 0, more than 1, 1 and less than 1. When we get the solution, we all know that during aerobic respiration, O2 is consumed and CO2 is released from the body. As we all know that, what is actually the ratio of CO2 evolved, or the volume of CO2 evolved from the body, is equal to the volume of O2, that is the oxygen consumed by the body. These both the values are of the equal, and these ratio is also denoted as the RQ. So RQ is what? RQ is the respiratory quotient. Respiratory quotient is the volume of CO2 evolved and the volume of O2 consumed in the body. When we discuss about the respiratory quotient depend upon the type of respiratory substrate that generally depend on the respiratory substrate that generally used during respiration, during the process of respiration. When we discuss about the first case, that is the carbohydrate, when carbohydrate are used as substrate and are completely oxidized in that case RQ is equal to and will be 1. RQ is what? The respiratory quotient, because in that case equal amount of carbon dioxide and oxygen evolved and consumed. What is consumed? Consuming of O2 and evolving of carbon dioxide, and it is also written as a RQ is equal to 6 CO2 and 6 O2, hence is equal to the 1. RQ in the case of carbohydrate is equal to 1. The next case is proteins. Next case is proteins. When proteins are the respiratory substrate, as we discussed in the earlier, that respiratory quotient will differ according to the respiratory substrate. We generally use the various kinds of substrate like the protein, carbohydrate, fat, organic acid. Along with that, the respiratory quotient value will also change. This is the case when protein are the respiratory substrate. The ratio would be about, that is the respiratory quotient is about 0.9 in the case when protein act as a respiratory substrate. Next is RQ, that is the respiratory quotient is slightly more than unity, slightly more than unity is found in when the respiratory substrate is organic acid, such as, because that work under aerobic conditions, for example is breakdown of oxalic acid, and it is also written as the respiratory quotient is when 4 carbon dioxide molecule is evolved and the 1 molecule of oxygen is evolved, then the respiratory quotient value is equal to the 4, and that is in the case of organic acids. This is the third case. In the next case, that is the case when fats are used as the respiratory substrate in respiration, in the process of respiration, then RQ, that is the respiratory quotient, is less than 1. For example, in the breakdown of tripalmitin, and it is also written as RQ, that is the respiratory quotient in the case of fat, is equal to the 102 molecule of carbon dioxide that is evolved, consumed, and 145 molecule of O2 that is evolved is equal to 0.7. That is the respiratory quotient is less than 1 and the value is 0.7, and that is in the case of fats. So according to the discussion, we just mentioned the case of carbohydrate, protein, organic acid, and fat. We generally check out the value for all that, and that generally should be in the series. In the carbohydrate, the value is equal to the RQ value is 1. We all know that respiration quotient value is 1. In protein is 0.9. Organic acid it's 4, and in fats the value is 0.7. So in the order in which they are demanding or asking in the question, according to that, the right answer for the question is The right answer for the question is option A, which show the sequential increase for all that, and the option 1, that is A, is the right one in this case. Let's discuss the next question from the chapter Respiration in Plants. The next question is, what is true about the end product of glycolysis? Option A, 2 pyruvic acid plus 2 ATP plus 2 NADH. Option B is 2 pyruvic acid plus 2 NADH. We all know that reducing power is known as NADH and ATP is known as the energy currency of the cell. And option C is 1 pyruvic acid plus 2 ATP plus 2 NADH. And option D is 2 pyruvic acid plus 1 ATP and 1 NADH. When we discuss about the actual process of glycolysis, in the glycolysis, 2 molecule of ATP are consumed during the 2 phosphorylation reaction. 2 phosphorylation reaction to form the fructose 1,6 biphosphate and in return of that 4 molecule of ATP are consumed. 4 molecule of ATP are consumed and produced. These 4 molecules are, except that 2 molecules which we consumed upper and the before for the phosphorylation, and in that 4 molecules are produced, that generally produced by the substrate level phosphorylation. And that phosphorylation is actually including the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate and phosphoenol pyruvate to pyruvate. Phosphoenol pyruvate to the pyruvate. During the process, 2 molecule of NADH are formed, and that are formed at the time of oxidation of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate to 3-bisphosphoglycerate. And if we can write the net reaction of glycolysis, net reaction of glycolysis, it is written as follows. The reaction is also written as when glucose add to the NAD plus 2 ADP plus 2H3PO4, and this is the reactant when convert into the or formation of 2-pyruvate plus 2NADH plus 2H plus 2 ATP. Why 2 ATP? Because in the process 4 ATP are produced, out of which 2 consumed, so we generally mention out the nutshell, that is the 2 ATP. And after the reaction, when we calculate the total product, even the NADH and the ATP, then we all know that NADH is equivalent to the 3 ATP. NADH is equal to the 3 ATP. So the net gain of ATP, that is the energy in the glycolysis is 8 ATP, or either written as a 2 NADH and 2 ATP. So according to the discussion, the right answer for the question is option A. Option A is the right answer in this case. Let's mark the right one in the option, that is 2 pyruvic acid and 2 ATP and 2 NADH is the right one in the case, as we discussed in the question. Let's discuss the next question from the respiration in the plants. That is, fate of pyruvic acid during aerobic reaction is option A, lactic acid or fermentation, B is alcoholic fermentation, C is oxidative decarboxylation, and D is oxidative phosphorylation. We all know that during the aerobic reaction and aerobic respiration, during the process, that is the aerobic respiration, the pyruvic acid which is formed during the process of glycolysis enter into mitochondrial matrix, and where it undergo the process named as oxidative decarboxylation to produce the two things from the pyruvate molecule, that is the carbon dioxide and NADH. Carbon dioxide and NADH, the product combined with the product that is formed by the oxidative decarboxylation combined with sulfur containing coenzyme A to form the acetyl coenzyme A. The reaction is catalyzed by enzyme complex named as pyruvate dehydrogenase. The enzyme name is pyruvate dehydrogenase, and the reaction and the step is also known as a link reaction or the gateway step. Link reaction or the gateway step, as it links the glycolysis to the Krebs cycle. And we can also write the reaction. For example, we just discussed the pyruvic acid when combined with the coenzyme A and the NAD plus in the presence of magnesium ions and the enzyme that is named as a pyruvate dehydrogenase. The reaction is acetyl coenzyme A plus carbon dioxide plus NADH plus H plus. This is the reaction which is generally asking in the question. So according to the discussion and the reaction, the right answer for the question is option C. Let's mark the right one in the question. That is the oxidative decarboxylation is the step and the reaction that generally occur in the process in which pyruvic acid is break down into the carbon dioxide and the NADH. So the right answer is oxidative decarboxylation is the right one in this case. Let's discuss the next question in the case. Next question is identify the A and B in the given reaction, where the reaction is write as a pyruvic acid plus coenzyme A and NAD plus in the presence of pyruvate dehydrogenase and the magnesium 2 plus ion. The product is A plus B plus NADH plus H plus. Options for A and B are given as A, that is the PEP and CO2, B is acetyl CoA and CO2, C is soy CO2 and H2O, D is acetyl coenzyme A and H2O. As we discussed, there is a reaction that is act as a, in the previous question, that is known as a link reaction or the gateway reaction. That is the link between the glycolysis to the Krebs cycle, and the reaction is, as we discussed in the previous question, that is the pyruvic acid plus coenzyme A plus NAD plus will give rise to the, in the presence of, as it is given in the equation, that is the Mg2 plus and the pyruvate dehydrogenase. The reaction is also given to the product acetyl CoA plus carbon dioxide plus NADH plus H plus. This is also the reaction and it is also come under the process that is the oxidative decarboxylation, where the pyruvate, that is the end product of our generally the glycolysis, is break down into the CO2 and NADH. So according to the discussion, the right answer for the question is option B. That generally denoted the same end product that is asking. Here the acetyl CoA is the A and CO2 is the B, is the right answer in this case. Let's mark the right one in the option, that is the B, that is A for acetyl CoA and B for the carbon dioxide. A here for the acetyl coenzyme A and B for the carbon dioxide is the right one in this case. Let's discuss the next question from the chapter Respiration in Plants. The next question is, as per Chemiosmotic coupling hypothesis, in the mitochondria, protons accumulate in the outer membrane, inner membrane, intermembrane spaces and the matrix. We generally discuss this with the help of, first discuss about the chemiosmotic coupling hypothesis. What actually this hypothesis is? That first of all, that hypothesis was proposed by the scientist named as Peter Michel in the 1961 and 66, and he got Nobel Prize for the same. In the year 1978. Sometime question is asking regarding their Nobel Prize taken and the discovery of the chemiosmotic and the proposal given by the which scientist. And this is the most widely accepted theory for oxidative phosphorylation. This is the most accepted theory for the oxidative phosphorylation. In the theory, they just discuss that reduced NAD, that is the NADH, is released from the Krebs cycle, and also transport 3 pairs of H plus ions across the inner mitochondrial membrane. They generally pass out it to the inner mitochondrial membrane, to the intermembrane space. As in the question, it is asking that where they travel, that is they travel actually to the intermembrane space. Similarly, NADH from that NADH come from the glycolysis, and FADH2 that generally come from the Krebs cycle. They also transport their protons, that is the H plus ion. They transport their protons in the intermembrane space. This is also known as a unidirectional flow of protons, that also work as the accumulation of protons in intermembrane space. Accumulation of proton in the intermembrane space, due to which there is a rising of the proton concentration gradient. because of the unidirectional flow of the proton in the intermembrane space, and due to which the pH of outer surface of inner mitochondrial membrane lowers as compared to the pH across the membrane. The pH of the outer surface of inner mitochondrial membrane lowers as compared to the pH of the of mitochondrial across the membrane, due to which the pH gradient and the proton concentration build a proton motive force, also known as a proton electrochemical gradient, across the membrane, and that is generally occur in the intermembranal space. So according to the question, the right answer for the question is option C. Option C is the right answer in this case. Let's mark the right one in the option, that is the intermembrane space, as there is the influx of the proton ions from the intermembrane, either from the glycolysis in the form of NADH and either in the form of FADH and NADH2 in the Krebs cycle. There is the generally influx of the proton ion from the intermembrane space. So intermembrane space is the right one in this case. Let's discuss the next question. The next question is, which of the molecule listed below is a product fermentation of a glucose by the yeast? Option A, C6H10O5 power N, C2H5OH, option C, C6H12O6, and D is CH3OH. When we discuss about the inalcoholic fermentation, pyruvate is converted into the ethanol, and that is generally the two-step pathway. Ethyl alcohol fermentation. First is ethyl alcohol fermentation. Ethyl alcohol fermentation is quite occur in the fungi. For example, in the case discussed in the Rhizopus and yeast. In the case Rhizopus and the yeast, and apart from the fungi, it is also occur in the some bacteria. First is the ethyl alcohol fermentation. Next is the in yeast pyruvate is decarboxylated to form the acetaldehyde, and which is then reduced by NADH to form the ethanol. And if we write the reaction in both the manner, in the first manner it is written as when pyruvate give rise to the, or generally give the carbon dioxide, and the reaction is occur in the presence of pyruvate carboxylase, pyruvate decarboxylate, it is converted into acetaldehyde. Along with the Mg2 plus ion, and in the second manner it is written as acetaldehyde give rise to the ethanol, and that generally take place due to the presence of enzyme named as alcohol dehydrogenase. So according to the question, the right answer for the question is option B. Let's discuss and mark the right one in the case, that is the option B, that is the C2H5OH, that is the ethanol is the right one in this case. Ethanol is right one in this case. So student, this is all about the respiration in plants. Hope student you like the lecture. Stay tuned with the Misostudy for such more interactive session. Till then, take care, stay safe at your places. Thank you.